Hello everyone, my name is Courtney and today we're going to be discussing the hydrophobic interactions between helix TM1 and helix H8 and how it is essential for proper rhodopsin transport and photoreceptors. Before we get started, there are a few things we need to go over. First is the main model organism used for this study, the African clawed frog, or Xenopus laevis, which I will refer to as Xenopus through this presentation. Also, we need to look at the eye. Do you see what I did there? Well, if not, then maybe the photoreceptors in your retina aren't working. Photoreceptors are the nerve cells of the eyes and they come in two flavors, rods and cones. The structure of these photoreceptors are similar. Here we see a rod. I highlighted the important structures for the sake of the article. First are the inner and outer segments. The outer segment is the segment that receives the light and is made of these free-floating discs. Light travels into the outer segment and into these discs, where it triggers rhodopsin, a light-sensitive GPCR, and a phototransduction cascade moves the signal into the inner segment and towards the optic nerve. Now, I keep on mentioning rhodopsin, but what is it and what does it do? This is rhodopsin, specifically bovine rhodopsin. Highlighted are two very important sections of rhodopsin, the transmembrane helix 1, TM1, and the helix 8, H8. Based on previous research, there are known hydrophobic interactions between TM1 and H8, so the researchers of this paper wanted to explore the hydrophobic interactions between these regions and their role in proper rhodopsin transport. Knowing that, let's get into the data. First, the researchers wanted to establish a basis for their study. They analyzed both wild-type and mutant forms of Xenopus rhodopsin, X-row, and zebrafish rhodopsin, Z-row, to compare how mutations at position 317 affected rhodopsin transport. Now, they chose position 317 because it is a highly conserved position in most species, but Xenopus rhodopsin is unique, having leucine at this position, whereas other species, including zebrafish, have methionine instead. To track rhodopsin localization, they've used GFP to the C-terminal domain on both X-row and Z-row for live cell imaging in receptors. As you can see here, when they mutated 317 in X-row from leucine to methionine, rhodopsin mislocalized and failed to reach the outer segment. Conversely, introducing the opposite mutation in Z-row, methionine to leucine, resulted in the opposite effect, improving outer segment transport. These results led them to hypothesize that position 317 on H8 and its interacting partner on TM1, M57, form a key interaction essential for proper rhodopsin localization. This is the same 317 mutation on X-row, but on full-length rhodopsin, not just the C-terminal. You can see some of the fluorescence of the mutated X-row here in the inner segment compared to none here. This mislocalization suggests that the mutation at position 317 disrupts rhodopsin transport efficiency, preventing it from reaching its correct destination in the photoreceptor cell. Both of these preliminary experiments suggest that the specific amino acid at position 317 plays a key role in rhodopsin transport efficiency and function in photoreceptor cells. Now, who can tell me an important characteristic of both leucine and methionine, the amino acids they used here? Yes, they are hydrophobic. So, with that in mind, what if, instead of swapping one hydrophobic amino acid for another, we swapped it with two different hydrophilic amino acids? Researchers replaced leucine at position 317 in X-row with arginine, a positively charged amino acid, or with aspartic acid, a negatively charged amino acid. When they did this, they noticed that changing from hydrophobic to hydrophilic residues created strong mislocalization, or incorrect transport of rhodopsin. This suggests that switching from a hydrophobic to a hydrophilic residue disrupts rhodopsin's proper transport, and that position 317 specifically is important in that. Let's take a look at the sequences and the rhodopsin structure to better understand this conclusion. Researchers looked at several rhodopsin sequences and found that the H8 helix interacted with the TM1 helix in several spots. Since their earlier experiments showed that hydrophobic interactions were important for rhodopsin trafficking, they hypothesized these interactions between H8 and TM1 might be important too. To test this, they wanted to block the hydrophobic interactions between the two helix. However, because there were multiple interaction sites and the 3D structure of X-row wasn't known, they needed to narrow down the key residues. They used an online tool called Vertebrate to create a model of X-row and then compared it to a known bovine rhodopsin structure. With this comparison, they could rule out some of the interaction points that were the same in both models. They did find that amino acids at positions 317 and 57 were swapped in Xenopus compared to other species. This suggested that rhodopsin transport depends on a broader network of specific residues, not one critical amino acid. Now, to actually test their theory. Building on what we've already learned, I pulled back in the leucine to arginine mutation on H8 to compare a similar mutation on its interacting partner on TM1. In both cases, replacing a hydrophobic residue with a hydrophilic one resulted in significant rhodopsin mislocalization, strongly supporting the idea that hydrophobicity, rather than a specific amino acid, is the key factor in proper rhodopsin transport. This finding reinforces their original hypothesis that hydrophobic interactions between H8 and TM1 are critical for rhodopsin transport in photoreceptor cells. The researchers also wanted to see if Xenopus rhodopsin was an exception or if the transport mechanism was consistent with other species. So they double mutated positions 317 and 57, swapping their amino acids to match zebrafish and bovine rhodopsin. Surprisingly, there was actually no significant difference in rhodopsin localization compared to wild-type rhodopsin. This suggests that the key factor in proper rhodopsin transport is the hydrophobic interaction between H8 and TM1, rather than the specific amino acids present. As long as the hydrophobic interaction is maintained between helices, rhodopsin localizes correctly, regardless of the specific amino acid present. This helps explain why rhodopsin transport works properly across different species, even with amino acid differences. Okay, so that was a lot, but what exactly is the takeaway here? First is that positions 317 and 57 both influence rhodopsin localization. Secondly, since these positions are interacting partners on H8 and TM1, that the hydrophobic interactions between the helices also influence rhodopsin localization. Third, as long as a hydrophobic interaction exists between helices, rhodopsin will find a way to help us see the bigger picture, literally. By studying how rhodopsin is localized, we get a clearer picture of how GPCRs, like rhodopsin, move within the cell and how other receptors might work. This kind of research is critical for developing new drugs, especially those aimed at vision-related diseases like retinal degeneration. Understanding rhodopsin's journey inside the cell also helps us spot potential treatments, not just for eye health, but for other neurological conditions tied to mislocalized receptors. Ultimately, this research has the potential to lead to better treatments and improved outcomes for patients. Thank you.